The aim of this procedure is to analyse Mycobacterium tuberculosis cell-free extracts by NMR metabolomics. First, culture and harvest the cells at an appropriate stage of growth. Then lyse the cells and obtain cell-free extracts for analysis. Resuspend the lyophilized extracts in buffer for NMR analysis. Finally, collect NMR data and analyze spectra with software to identify metabolites. Ultimately, NMR analysis of Mycobacterium tuberculosis is used to show perturbations in metabolite levels upon various treatments.
NMR metabolomics can be applied to key aspects of the physiology of Mycobacterium tuberculosis, such as important metabolic pathways. This understanding is critical to elucidate the mechanisms of drug action and resistance and to identify novel targets for drug design.
Generally, individuals new to this method would struggle because of inability to handle all samples exactly the same, which will introduce unnecessary variations in results. From my laboratory, Stephen Halouska, a PhD graduate student, will demonstrate the procedures to handle the samples for NMR processing.
From my laboratory, Denise Zinniel, Research Laboratory Manager, and Robert Fenton, Research Technician 3, will now demonstrate the procedures to handle Mycobacterium tuberculosis cultures.
When conducting M. tuberculosis research, follow Biosafety Level 3 practices as defined by institutional guidelines. Although the labels in this video say Mycobacterium tuberculosis, for biosafety considerations, this demonstration used the non-pathogenic